My presentation is about analytical characterization of lead structures, pharmaceuticals, and biopharmaceuticals. Upon completing this lecture, students will be able to emphasize the importance of instrumental analysis for the characterization of both pre-approved and post-approved pharmaceuticals and biopharmaceuticals, to identify the need for analysis prerequisites and sample preparation methods, and to have an overview about some instrumental techniques which are used in pharmaceutical analysis. Do you know that good analytical characterization offers better drug performance? And that proper analytical technique is the key for pharmaceutical research? In this presentation, I'm going to give you an overview about the use of analytical methods in drug characterization. I have divided my presentation into two parts. First, I'm going to talk about analysis in drug discovery, development, and production, where I will go through 3 points: characterization, scope of analysis, and analysis prerequisites. And in the second part of my presentation, I'm going to give you an overview about the use of 4 selected analytical techniques in drug characterization, namely liquid chromatography and mass spectrometry, capillary electrophoresis, high-resolution continuum source atomic absorption spectrometry, and microscale thermophoresis. So first, about characterization. Characterization as a general term refers to the study of drug structure, activity, stability, potency, behavior in formulation, and behavior in vivo. When we are speaking about analytical characterization, then we restrict the term to the use of analytical methods to study the structure and properties of a substance. Now, about scope of analysis. Here I would like to talk about pre-approved pharmaceuticals and biopharmaceuticals. Usually, the early discovery and development of a small drug molecule is referred to as new chemical entities, which can end up with a pharmaceutical, while the early discovery and development of a protein-based molecule is referred to as new biological entities, which can end up with a biopharmaceutical. And here you can see the difference between pharmaceuticals as aspirin and biopharmaceutical as a monoclonal antibody in terms of size. The process of finding a new drug includes drug discovery and development. Drug discovery involves 3 stages: target identification and validation, lead identification, and lead optimization. During target identification and validation, we do need analytical methods to study proteins, that is to say, to do proteomics, and to study genes, that's to say, to do genomics. But also during lead identification, we need analytical methods to study, to follow up the synthesis, the isolation, and the study of the physicochemical properties. And also during lead optimization, we do need analytical methods for quantification, for stability testing, and for bioanalysis. Not only during drug discovery, but also during the drug development, the next stage of finding a new drug, which includes preclinical trials and clinical trials, we do need analytical methods. For example, during preclinical trials, to study the ADME, the absorption, distribution, metabolism, and excretion, and acute short-term toxicity, and also during the clinical trials to do bioanalytical studies to evaluate the ADME and the dose and the toxicity. So what about after approval? Do we still need analytical methods for approved pharmaceuticals and biopharmaceuticals? The answer is yes. Analytical methods are required for approved drug, for example, to study their bulk drug materials, to check their quality, for intermediate of drug synthesis to follow up the synthetic process, for drug pre-formulation to check their compatibility, for drug impurities to ensure the absence of impurities, for drug degradation product to check stability, and for cleaning validation to ensure good manufacturing practice. This is why one can say that a proper analytical method is the key for pharmaceutical research. Now I come to analysis prerequisites. In general, before starting the analysis, one should have a knowledge about the structure of the substance and initial knowledge about physicochemical properties like charge, molecular weight, and solubility. The analyst should then consider the selection of a proper analytical technique. But depending on the aim of analysis and the complexity of the sample, the analyst should consider three important aspects before starting the analysis. These aspects are the possible need for a sample preparation technique to isolate the target analyte from the matrix, the possible need for a separation method to separate the target analyte from other components in the mixture that may interfere with its analysis, the selection of a proper detection method to detect the analyte. Especially when dealing with biological samples like blood, plasma, urine, cerebrospinal fluids, and other with high percentage of matrix, sample preparation should be considered. If sample preparation is needed, the analyst should select one of the four main sample preparation methods. These are liquid extraction, protein precipitation, solid phase extraction, and a method called dilute and shoot. Now I come to the second part of my presentation and would like to briefly introduce four selected analytical techniques which I usually use in drug analysis. These techniques are one of the chromatographic techniques, namely high-performance liquid chromatography, usually coupled to mass spectrometry, one of the electrophoretic techniques, namely capillary electrophoresis, one of the spectrometric techniques, namely high-resolution continuum source atomic absorption spectrometry, and one of the thermophoretic techniques, namely microscale thermophoresis. So first about liquid chromatography and mass spectrometry. Here I would like to briefly explain the technique. If you have two or more analytes in a mixture and you would like to analyze them without interference, then you can use this technique. The separation takes place between two phases. One is a mobile phase and the other is a stationary phase. The substances will move with the mobile phase over the stationary phase and will make interaction with the stationary phase. Because they are of different structure, the interaction will be of different strength. So it could be strong for one analyte and weak for another. And so they will move with different speed and will separate from each other. They will reach the detector at different times and will be detected at different times, and the result will be a peak or a signal that we call a peak. Currently, there are three competing approaches for fast HPLC. These approaches are the use of monolithic silica columns, the use of fused core particles, and the use of ultra-performance liquid chromatography. I have extensively worked with monolithic columns and published several articles, one chapter in a book and a book describing their use for fast analysis, also using flow programming for bioanalysis and for the separation of complex analytical mixtures. Here you see an application example which my PhD students and I conducted for the separation and the quantitation of 5 anti-diabetic drugs by liquid chromatography mass spectrometry. Now about the second technique, the capillary electrophoresis. In fact, there are different instruments from different companies, but the principle is the same. The principle is that particles with different electrophoretic properties have different migration speeds in electric field. If we consider the standard capillary electrophoresis method, which is the capillary zone electrophoresis, The separation takes place according to mass to charge ratio. Here you see the separation order for different ions. This technique can be used to investigate protein-ligand interaction using a method called affinity capillary electrophoresis. Here you see an application example to investigate the interaction between human serum albumin and heparinoids. The third technique is high-resolution continuum source atomic absorption spectrometry. High-resolution continuum source atomic absorption spectrometry is the most innovative technique in the field of atomic spectrometry. The instrument was produced by a German company called Analytical in 2007, and the instrument can effectively and simultaneously determine several metals in the sample with high sensitivity. Here I have used the instrument to study the uptake of nickel-methoxysilophane complex, a potential anti-cancer drug, by breast cancer cell line, and here also to study the uptake of the complex by a colon carcinoma cells. Now I come to the last technique, the microscale thermophoresis. In fact, microscale thermophoresis is a relatively new technique introduced by a company called Nanotimber from Germany and can be used to study molecular interaction based on migration in a temperature gradient. When subjected to a heat through a laser, the molecules will migrate away from the heat, so the fluorescence rate will drop. If the process is repeated in the presence of ligand, the speed by which the molecules escape from the heat will be slower, so the drop in fluorescence rate is less. If we repeat this process in the presence of different concentrations, we can generate a binding curve which gives us information about the binding strength. In this example, my PhD students and I use this technique to investigate the binding of tetrapiron to different metal ions. As a key message, analytical characterization is mandatory before and after drug approval. Analysis prerequisites should be considered before starting the analysis, and different instrumental techniques can be used for drug characterization, but the overall aim is to improve drug performance.